Hello and good morning, everybody. Welcome to GemPharmatech's monthly webinar series. My name is John Jackson. I'm part of the marketing team here at GemPharmatech. And today we're going to hear from Dr. Yueshun, a study director from the cardiovascular team at GemPharmatech, as she explores the topic genetic mouse models for hypertrophic cardiomyopathy, HCM, and the models MYBPC3 knockout and the MYH6 R404Q model. Before we do that, I'm going to take a little bit of time to introduce GemPharmatech and what we do. We are a preclinical CRO, and we are trying to advance science and engineer a healthier world by pushing the boundaries of biomedical innovation. The way we do that is by providing the models, data, and insights that move drug discovery forward. Central to this idea, and really central to the foundation of GemPharmatech as a company, was and is this unrivaled library of proprietary in vivo models. This includes over 30,000 genetically engineered models, around 22,000 of which are knockout models, part of the Knockout All project, which was developed to give scientists, unrivaled access to tools that they might need for a specific target gene of interest. And we have a goal to knock out all of the protein coding genes in the mouse genome. We have the capacity to generate 6,000 new strains each year. And as we continue to work with biotech and big pharma on different preclinical studies and drug efficacy testing, we conduct over 1,000 of these studies each year. And I think that number has actually grown over the last year already. Now, today, Yueshun is definitely going to explore our cardiovascular services and models, but just a reminder that we work across disease areas, indications, modalities. So if you are interested in hearing more from us, I encourage you to follow us on LinkedIn. Definitely be on the lookout for more webinar invites. And if you're going to events, we definitely are active in the event and conference scene. So we post on our LinkedIn where we will be, and you can come talk to us and meet us. With that, I will leave it to Dr. Yueshun to explore this topic. At the end, we're going to have a Q&A session, and we're going to use the Q&A module on Zoom. If we don't have a chance to get to your question, we'll definitely reach out to you after the show. You can reach out to us on LinkedIn as well. So with that, thank you, and Dr. Yueshun.
Okay, thank you, John. Good morning, everyone. Nice to meet you all. So today I'll present our work on hypertrophic cardiomyopathy. So today's presentation is composed of four main parts. So first, I'll give a brief introduction to HCM-associated gene mutations. Second, then I will provide an overview of the phenotypic data we currently collected on these two genetically engineered mouse models, which are mybp3 knockout mice and MYH6 point mutation mice. And then I will also provide some efficacy test results we have on these two animal models. And lastly, all the data includes two key cardiac function assessment tools, which are echocardiography, which provides functional and structural imaging, as well as PV loop catheterization, which provides hemodynamic parameters measurements. So these two complementary tools help to provide a more comprehensive cardiac function evaluation in vivo studies. So first, hypertrophic cardiomyopathy, short for HCM, is a disease characterized by the thickening of heart muscles, particularly the ventricular septum. So this thickening will make the heart stiffer, which in turn impairs its ability to reflex as well as filling properly during diastole, and this will ultimately leads to or impairs the pumping efficiency of the heart. So from the diagram here, we can see there are two major different types of HCM, which are concentric hypertrophy. This is characterized by increased left ventricular wall thickness, as well as a decreased internal diameter of the left ventricle. This is more associated with heart failure with preserved ejection fraction, HFpEF. And the other type is eccentric hypertrophy, characterized by increased internal diameter of the left ventricle. And it's a decompensated state with reduced systolic function. So this is more associated with the half-ref phenotype. So most of the HCM cases, or specifically 55% of the HCM cases, are associated with sarcomeric gene mutations. The two most dominant genes are MYBPC3 and MYH7 in human. So we can see this figure here. MYH7 and MYBPC3 both accounts 17.5% of all the HCM cases. Interestingly, the mutation patterns between these two genes differ greatly. For MYBPC3, more than 70% of the mutations are premature termination codons, which produce truncated protein or protein loss. And for MYH7, more than 90% of the mutations are missense. This produce the full length but dysfunctional protein. So therefore, our GemPharmatech generated these two genetically engineered mouse models to recapitulate these two HCM subtypes. So this slide gives an overall information of these two HCM models. So first, we see the MYH6 point mutation mice. We use a knocking strategy which carries the heterozygous point mutation, and this mice starts to exhibit concentric hypertrophy with improved systolic function after postnatal 8 weeks in both female and male mice. And this model starts to show fibrosis around 6 months of age. And for this mouse strain, we have tested a lot of studies using mavacamten and the small compounds, as well as ABE gene therapy. In comparison, for the MYBPC3 knockout mice, it starts to exhibit the eccentric hypertrophy with impaired systolic function as early as postnatal 2 weeks. And also we can observe fibrosis as early as 3 to 6 weeks of age. So for this mice strain, we have also tested a lot of AAV-mediated MYBPC3 replacement test articles and also some other small compounds. So first I'll talk more about the MYBPC3 mouse strain. So first I will talk about the haploinsufficiency of the MYBPC3 resulted HCM. So here in the normal heart, in the normal sarcomere, the MYBPC3 cross-links with actin and myosin to regulate calcium-driven contraction of the heart. However, so in normal cases, these sarcomere proteins will constantly synthesize and be degraded to maintain a balance. However, after the MYBPC3 mutations, this will produce a reduced protein level, which will disrupt this balance and impairs the contractile function of the heart. So basically, the MYBPC3 resulted HCM is resulted from a too little functional protein. Therefore, we developed this MYBPC3 knockout mice. So these mice can be used for anti-HCM drug screening and efficacy test, especially gene therapy replacing human MYBPC3 into cardiomyocytes, and also can be used for diseases such as hypertrophy and ventricular dilation or fibrosis caused by the defects of MYBPC3. So here the Western blot data shows the ablation of the MYBPC3 protein in the mice. We can see the ablation of the MYBPC3 protein in both female and male mice. So here shows the echocardiography data of the mybp3. Since the mice start to exhibit the phenotype eccentric myocardial hypertrophy as well as systolic dysfunction as early as 2 weeks of age. So we have the data from 2 weeks of age until around 40 weeks of age. We can also perform the drug administration through retroorbital vein as early as 2 weeks of age. So first, let's see the echo data. We can see as early as the 2 weeks of age, the left ventricle septum thickness is significantly increased compared to the B6 groups. And over time, it's also continually increasing. And also, we can see the left ventricle post-wall thickness. It's also continually increasing over time. Same as the left ventricular and the diastolic volume. Importantly, we can see the injection fraction here. It's significantly reduced over time as well. So this might display the eccentric myocardial hypertrophy as well as systolic dysfunction. So this slide shows the pathological analysis for this mouse strain at postnatal 2 weeks and 3 weeks and 5 weeks. So here shows the HE staining and the PSR staining data. So we can see the enlargement of the left ventricle chamber as early as 2 weeks of age. And the 3 weeks of age, we can see the fibrosis to show. And for the 5 weeks of age, we can also see the PSR staining here, the fibrosis. So here shows the pathologic analysis of this mouse strain at 3 months of old. So compared with the wild-type mice, the hearts of the mybp-C3 mice were significantly enlarged. And also higher heart weight to body weight ratio is preserved in both male and female mice. And also the HE staining showed eccentric cardiac hypertrophy both in the male and female mice. And here shows the myosin staining data, and the mice start to display the fibrosis at 3 months of age. So then is the second animal model, which is Myh6 R404Q point mutation mice. So MYH7 is the predominant myosin heavy chain isoform in adult human ventricles, and MYH6 predominates in the adult mouse ventricles. So structurally, mouse MYH6 is equivalent to human MYH7, and they share high sequence identity. So in this mouse strain, we use a knocking strategy to introduce a heterozygous point mutation. So this animal model can be used for anti-familial HCM drug screening and efficacy tests, as well as for research on related disease such as myocardial hypertrophy and fibrosis caused by defects of the MYH6. So here we can see this shows the MYH7 human gene sequence as well as MYH6 mouse gene sequence. They show high sequence identity. So the strategy here is to replace the R arginine amino acid with Q glutamine. So here echocardiography data shows the B6 MYH6 point mutation heterozygous mice showed the concentric cardiac hypertrophy with increased systolic function phenotype. So we can see first the left ventricular posterior wall thickness from 6 weeks of age it's continually increasing, and as we have the data until 1.5 years of this mice. These mice can survive until around 1.5 years. So yeah, we can support long studies for this mouse strain. And also for the IVS, left ventricle septum thickness, it's also significantly increased compared to the B6 mice. As well as we can see the left ventricle internal diameter, it's increasingly decreasing. It's continually decreasing. And for the injection fraction, starts from the 8 weeks of age, it starts to show significant increase compared to the B6 mice. So overall, it shows a concentric cardiac hypertrophy with increased systolic function phenotype. We also have tissue Doppler data to measure to assess the diastolic function of this mouse strain. So here we can see the E prime value, which reflects the innate cardiac relaxation capability. So at 16 weeks of age, it's significantly lower compared to the B6 mice, and also the E to E prime ratio at 6 weeks of age is also significantly higher, and also the IVRT, which is measured through tissue Doppler imaging, it's significantly increased compared to the B6 mice. So overall, these mice also showed the moderate diastolic dysfunction after birth 16 weeks. We also have collected PV loop data on this mouse model. So consistent with the echo data, PV loop data also showed that first we see the dB/dt minimum value. It shows a significantly reduced absolute dB/dt minimum value, as well as significantly increased tau value. This all means this mouse strain shows impaired diastolic function. And also after IVC occlusion, we get the ESPVR and EDPVR value. So ESPVR, here, the significantly higher ESPVR reflects the increased systolic function of this mouse strain. And also the significant increased EDPVR value reflects the impaired diastolic function of this mouse strain. So compared with the mYBPC3 knockout mice, which shows fibrosis as early as 2 weeks of age. So this mouse can only exhibit fibrosis or is interstitial fibrosis or replacement fibrosis have to be around 6 months of age. So from this 8-week to 10-week-old HE staining and PSR staining, we don't see any significant increase in the fibrosis. Here is the pathology analysis of the MYH6 mice at 6 months of age. So at this time, we can see the significant increase in the fibrosis, both in the male and female mice. So in this model, we've tested the positive control drug, which is mavacamten. So here shows the x-axis means hours post-administration. It shows that after the administration of mavacamten, around 1 hour, the ejection fraction and fractional shortening was significantly reduced. And then after like 24 hours of the administration, the ejection fraction returns to the baseline level. So this helps us to determine the dosage as well as the dosing frequency of mavacamten. So this here, the efficacy data shows the daily administration of mavacamten for 8 weeks. And also we can see the data here in terms of the left ventricle septum thickness. It's significantly reduced after 8 weeks of the mavacamten administration. And also for the left ventricular wall, posterior wall thickness, it's also significantly reduced. For the left ventricular internal diameter at systole, it's also significantly increased. And also we can check the ejection fraction after the mavacamten administration. The ejection fraction was reduced. So here, this table summarizes the phenotypic, the pathological feature of the MYH6 point mutation mice. So it starts to show the sarcomere disarrangement, and the interstitial fibrosis has to be around 4 to 6 months of age. And for the replacement fibrosis, it's around 6 to 9 months of age. And to do the we can also perform immunofluorescence staining and electron microscopy to quantify and visualizing the sarcomere disarrangement. The common sarcomeric markers we use is alpha-actinin, actin, myosin heavy chain, and titin. So we are able to perform this staining to visualize the sarcomeric disarrangement. So besides these two HCM mouse models, MYH6 point mutation mice and MYBPC3 knockout mice. We also have other HCM and DCM mouse model in development, like RBM20 point mutation mice, and PCP2 conditional knockout mice, and NAMNA N195K mutation mice. So this mouse is also kind of popular these days, but it's still under development. It's a typical model for the DCM. And also we have the BAC3 conditional knockout mice. So these four cardiomyopathy models are still under development, but the phenotypic data will be available soon. Yes. So that's all for my presentation. Do you have any questions?
Thank you, Dr. Yueshen. We have quite enough time to answer anybody's questions. So, okay, we have one here. Your data demonstrated that mavacamten improved systolic function in MYH6 mice. I'd be interested to know whether a similar improvement was observed in diastolic function. In addition to the echocardiography findings, do you have any PV, I think they mean pressure volume, loop data to further support this?
Yes, we do have the mavacamten data on diastolic function on the MYH6 mice, but it's not internal data, so we are not able to provide the data here. But mavacamten did improve the diastolic function of the MYH6 mice, and also both in the echo data as well as the PV loop data. Yeah.
Thank you, Dr. Yashan. If you have any follow-up questions, feel free to also pop those in the chat. Another PV related question. Could you please clarify your daily throughput for PV loop measurements? Additionally, are the staff performing their cardiac assessments, echo, PV loops blinded?
Oh yeah, currently for PV loop daily throughput, I think now we are capable of performing 7 to 8 mice per day, and yeah, definitely our technicians are blinded to the group information. Yeah.
Thank you. Dr. Yashan, do you see the questions there in the box? Do you want to
Yeah.
pick one?
Okay. Does GemPharmatech specifically on cardiac macrophages related knockouts on stromal cells tracer models? Currently we do not have these models, cardiac macrophage related knock. No, sorry, currently we don't have, but we can talk in detail later and give you some feedback on this. Thank you.
Since the MYH6 mutant mice are on a B6 background with slow progression, do you have plans to test this in other strains, maybe to speed up the phenotype?
Yeah, we do have since a lot of clients are asking about this because the disease progression of the MH6 is kind of slow. We are currently crossing the B6 MH6 with FVB background because some papers reported that it can start to exhibit the phenotype as early as 4 weeks of age and will show fibrosis around 2 months of age. Yeah. So we are also collecting internal data on that part. Once it's available, we will publish the results on our flyers. Thanks.
Looks like there's kind of a related question also about the MYH6 point mutation model. Have you generated any efficacy data for aficamten?
Yeah, aficamten is still in the phase 3 clinical trial, but it shows better PK profile than mavacamten. So yeah, a lot of clients are also asking about this question. So we are also collecting internal data on that part. But from the systolic function, we have collected some data. It does truly improve the systolic function of heart after the aficamten administration. Thanks.
Thank you, Dr. Yoshen. Oh, when will the LMNA N195K DCM mouse be available?
It's we have the strategy ready, and it's still in the development phase. I think it should we will have the mice around August, and then after 2 months of the phenotype data collection, I think we'll have the data ready around October. Yeah.
Thank you. Is there any last questions before we sign off? I will say to everybody that, as the company with the largest collection of genetically engineered mouse models and a capacity to generate 6,000 each year, we're certainly very skilled in custom model development. So if you are interested in generating a novel model, please reach out to us to discuss that as well. Looks like there's no more questions. I will thank everyone again for attending. Again, please follow us on LinkedIn. We do these webinars on a monthly basis, and we explore topics across disease indications and modalities, as I said at the beginning of the presentation. Um, Looks like there's no more questions, so we'll sign off here. Thank you, everybody, and thank you, Dr. Yueshen.
Thank you.